So hello everyone, we will study transformation today. Okay, we will study transformation. So what is transformation? Transformation is basically a type of genetic recombination. It occurs in prokaryotic cells such as bacterial cell and archaeal cell. Okay, so what is transformation? Transformation is the uptake by the cell of DNA. That DNA can be either a plasmid DNA or a fragment of linear DNA. So it is basically the uptake by cell of DNA, and where the DNA is present, so of DNA from the surrounding, and its maintenance in recipient in a heritable form. It was discovered by Griffith in 1928. So let's understand what is transformation. Suppose there is a bacterial cell, okay, and in the environment DNA is present. This DNA can be a fragments of linear DNA or either a plasmid DNA. So what will happen in transformation, that this there is uptake of the DNA present in the surrounding environment by the recipient cell. Okay, and now this DNA, this upcoming DNA is known as exogenode, and the bacterial's chromosomal DNA is known as endogenode. Okay, and now what happens, the exogenode, that is the foreign DNA, now will recombine with the host's with the recipient's chromosome, that is with the endogenode. Okay, via homologous recombination. So what is transformation? It is basically the uptake of DNA by the cell. Uptake of DNA. Okay, now bacteria can uptake the DNA naturally, okay, without any efforts made by us. Okay, so this is natural transformation, in which bacteria naturally take up the DNA from their surrounding. However, there are certain bacteria that can not do this naturally. So this is artificial transformation, that they cannot uptake the DNA naturally. So in the lab they are forced to do so. Okay, so they are forced to be competent artificially. So this is artificial transformation, in which bacteria are made competent artificially by various means, and this occurs in laboratory. Okay, so what are the methods used in artificial transformation? So there are some methods. Okay, first is using a divalent cation along with heat shock. Divalent cation, for example, calcium has a valency of +2, so it is a divalent cation. Okay, when we use divalent cation along with heat shock, okay, this is one of the method used in artificial transformation. It is suitable for bacterial cell. Now next is electroporation, in which we use weak electric fields. Okay, this creates pores in the bacterial cell envelope. Through these pores the DNA enters the cell. Okay, micro injections and ballistics or gene gun, this is another way. Micro injection is suitable for animal cell, and gene gun, also known as ballistics, is suitable for plant cell. Okay, and now the last method is using PEG. PEG stands for polyethylene glycol. Okay, this is a chemical, and this can also be used to make bacteria artificially competent. Okay, so let's see the example of a bacteria which is naturally competent, which can be transformed naturally, and a bacteria which cannot be transformed naturally. They need to be made competent artificially. Okay, see example, Streptococcus pneumoniae. Okay, Streptococcus pneumoniae is naturally competent. Okay, means they can take up the DNA from the surrounding naturally without any efforts made by us, whereas E. coli is not naturally competent bacteria. It has to be made competent artificially in the lab. ठीक है? Okay, now we are using the term competent. So what does it mean? So competency is the ability of the cell to take up the free DNA. Okay, and the cell is known as competent bacteria. If the cell can take up the DNA, then the cell is known as competent bacteria. If the cell cannot take up the free DNA, it is known as non-competent bacteria. Non-competent bacteria are made competent using artificial transformation means using various methods. Okay, so what is transformation frequency? Transformation frequency is basically the proportion of the cell that gets successfully transformed. Let's suppose there are 100 cells. Okay, let's suppose there are 100 cells, and I put those 100 bacterial cell on a petri dish. Okay, the petri dish also had several DNA fragments. Okay, now what happens? Out of that 100 cell, only 2 cells, let's suppose 2 cells were naturally competent. Okay, so what happened, that those 2 cells took up the DNA. Okay, now the DNA, that is the exogenome, is present in the cytoplasm, in the cytosol. Now what happens when the DNA enters the cell? Let's suppose in one bacteria that DNA gets degraded. Okay, it does not got homologously recombined. Okay, however in another bacteria it got homologously recombined. So when we talk about stable transformation or successful transformation, so we can see that out of 100 cells, 2 cells took up the DNA. However, only 1 cell got successfully transformed. So when we write transformation frequency, we will write 1 divided by 100, that is 0.01 is the transformation frequency. It means that out of 100 cell, only 1 cell gets successfully transformed. The another cell took up the DNA. However, transformation did not occur, successful transformation did not occur. Okay, because it did not integrate into the bacterial's recipient's chromosome. Okay, now let's see the steps involved in transformation. So what happens, see what we studied in transformation till now, that there is DNA in the environment and the bacteria uptake the DNA. So where does the DNA comes from in the environment? Okay, so the DNA come from the lysis of the bacteria. Okay, first the bacteria lies and it releases its double-stranded DNA into the environment. Okay, and now this DNA has to be taken up by the recipient bacteria. Okay, so first step is the donor bacteria lies and releases its double-stranded DNA into the environment. Now the question arises, why the bacteria lies? So there could be various reasons for this, like there could be a viral infection and the viral performed a lytic cycle and the bacteria lysed. Okay, or there could be a hypotonic solution. Okay, and the bacteria lysed. So there could be various reasons. However, our main focus is that the bacteria should lyse, then only it will release its double-stranded DNA into the environment. Now there is double-stranded DNA in the environment and this DNA has to be uptaken by the cell. Now how this will occur? So the next step is the double-stranded DNA now binds to the competent cell through the receptors. Okay, and extracellular endonuclease cuts the DNA into smaller fragments. Okay, see what happens, the recipient chromosome has receptors on its cell surface. Through these receptors this double-stranded DNA binds. Now this DNA can be very long and this long DNA cannot be taken up by the cell. Okay, so there are some extracellular endonuclease enzyme. What these enzymes does is they cut this DNA into smaller fragments for the easy uptake of the DNA. Okay, now always remember that in transformation only single strand of DNA is taken up by the cell. Okay, and but the double-stranded DNA binds to the cell surface. Now the double-stranded DNA has bound and the single-stranded DNA has to be uptaken. So one strand has to be degraded. Now the next step is the one strand is hydrolyzed or degraded and the remaining single strand is transported into the cytosol by an uptake system. Okay, so one strand will be degraded or hydrolyzed and the remaining single strand will be taken into the cytoplasm by an uptake system. So there is a presence of uptake system in the cell envelope of the bacterial cell. Now see the next step. We will also see the diagram later on. Okay, so see, the next step is the exogenode, that is the chromosome DNA that has to be taken up by the cell. This is known as exogenode. Exogenode aligns itself with the homologous region on the bacterial chromosome. Okay, and homologous recombination occurs and heteroduplex DNA forms. See, I have to go on see white page to make you understand this. See what happens. Let's suppose this is bacterial's, this is bacterial recipient bacterial's chromosome. ठीक है, okay, it is double stranded. Okay, see, okay, and this is the foreign DNA. Okay, now what happens, suppose it has a sequence A A A A A A A A A. It has this DNA sequence A. Okay, and this has sequence A T A. Now what happens, that these are homologous chromosome because they have similar sequences. Homologous chromosomes are those chromosomes which have similar sequence. Keep in mind, this is not necessary to have same sequence, similar sequence are there. ठीक है, it has A. It also has A. Yeah, it has one T. However, except this part, all sequences are same. So all sequences are similar. So these are homologous chromosome. Okay. Now what happens, that this DNA will align itself to the homologous part. So let's suppose it will align. It aligns here. It aligns in this region. Okay, and now it will do homologous recombination. So it will do homologous recombination. So it will look like this. This is the second strand, and this is A. This is the bacterial chromosome that homologously recombine, and this is the other part. Okay, and this is A T A. Okay, now this homologously recombined. However, it has TT all around. Okay, it has TT all around. Okay, now what happens, that all the regions are homologously recombined except this region. So here a heteroduplex DNA forms. Okay, heteroduplex DNA forms. It occurs like this. This is heteroduplex DNA. Okay, now let's come back. This heteroduplex DNA forms, and now heteroduplex DNA is repaired. So what is heteroduplex DNA? Heteroduplex DNA is DNA in which both strand comes from two different sources and both strands are not perfectly complementary at some position. I have already explained you. Now this heteroduplex DNA is repaired. How will what does it mean? See what happens, this T will be removed and A will be inserted. Okay, now it will perfectly recombine. So heteroduplex DNA is repaired, so it will appear like this, normal. Okay, this is repair of heteroduplex DNA. Okay, now let's see the figure. See, this is a bacterial cell. Okay, and it has bacterial chromosome, its own chromosome, and in the surrounding various DNA fragments are there. What happens, the uptake of DNA, and the DNA is uptaken in single stranded form. So the bacteria has uptaken this DNA. However, it can do homologous recombination or it can be degraded, okay, by the host immune system by a restriction endonuclease. So if this happens, then it is unsuccessful transformation. But when it does homologous recombination, it recombines with the bacterial chromosome. Okay, and this is stable or successful transformation. So this was a transformation with the linear DNA. There could be transformation with the circular DNA like DNA plasmid. So there's DNA plasmid in the environment and the uptake of the plasmid, this, and this is stable transformation. If this plasmid is degraded, then it should be unstable or unsuccessful transformation. Okay, now see, we will see the transformation diagram in detail. See, this is bacterial chromosome. Okay, recipient chromosome. Let me focus. Yeah, this is bacterial cell having bacterial chromosome, and this part of the bacterial chromosome has lac minus gene. What is lac minus gene? Lac minus gene is that gene which does not synthesize proteins for the metabolism of lac, that is lactose. Okay, this lac minus gene is called so because it does not synthesize protein which is involved in the metabolism of lactose, so it is lac minus gene. And let's suppose in the environment there was a presence of a DNA which was having lac plus gene. Now this part of the DNA will recombine with the will attach with the cell surface with the help of receptors. Now this DNA is very large, it cannot be and it cannot enter the cell. This whole long DNA cannot enter the cell, so this DNA has to be degraded at some parts so that it could be converted into smaller fragments for the easy uptake. So there are extracellular endonuclease which cuts the DNA into smaller fragments. See what happens, this is lac minus gene, yes, and the extracellular endonuclease cuts the DNA and the lac minus gene attaches the receptor. Now what happens, we know that single strand is taken up, so one strand is degraded and single strand is transported by an uptake system. Okay, so this is an uptake system and single strand is going inside the cell. Okay, now see, DNA strand aligns itself with homologous regions on bacterial chromosome. What happens when the DNA enters? Now it will align itself with the homologous region. Okay, this was lac plus gene and this is lac minus genes, so both are having similar sequences, so these are homologous regions, so it will align itself here. Now what happened, DNA strand is incorporated into bacterial chromosome by homologous recombination. Now DNA strand will do these both DNA will homologously recombine with each other. Okay, and a heteroduplex region is formed. Now this heteroduplex region is repaired. Heteroduplex region is repaired in a way that changes lac minus strand to lac plus gene. Okay, see, this was lac minus part, and the lac plus and the black, actually see here, this blue part is lac minus region and the black strand is lac plus. Okay, now what happens, that the blue DNA will also become lac plus. Okay, this is transformed cell. Okay, so this is transformation. I have made these notes from Prescott's microbiology. If you know that Prescott microbiology is one of the best book to study microbiology. Okay, so if you will write this much in your exam, okay, you will get full marks. Okay, thank you.